Hi students, this is Professor Schimmold and I'd like to talk to you today about microbial metabolism. And guys, we're going to keep this really simple, so all of you chemistry freaks out there, don't get too upset when you see me simplify some of the concepts here. Okay, so let's take a look at your handout. And it starts out with some definitions of catabolic and anabolic reactions. Now, first of all, let me define metabolism. And the term metabolism refers to the sum of all chemical reactions that occur within a cell. And those reactions are divided up into two categories. Catabolic reactions, those are reactions that break larger molecules down into smaller ones, and in the process we're going to release energy that was in the form of chemical bonds, and then we can use that energy for a variety of tasks to synthesize other molecules, for cellular growth, for reproduction, anything else you can think of that a cell might need to do. And then anabolic reactions, they actually require energy, they consume energy, and we're going to use anabolic reactions to take smaller pieces, smaller molecules or atoms, and assemble them into larger structures, molecules such as, for example, DNA and RNA and complex proteins. Okay, let's see, where are we? I'm looking at the first page of your handout, and as it says there, the energy produced in catabolic reactions is going to be used to fuel anabolic reactions. And the energy that we're going to produce is going to be stored in the molecule known as ATP, adenosine triphosphate. Now, if you take a look at that molecule, and there's a diagram in your handout, and if you take a look at that, and I just drew part of the molecule on this page right here, and so you've got your carbon rings over here. What I wanted to show you was this is what we refer to as the terminal phosphate group, all right? So it's at the end of the ATP molecule, and this part of the molecule can either be removed, and when it's removed, then we have formed a molecule of ADP, diphosphate, or it can be added back to an ADP molecule, in which case we have made a molecule of ATP. Now, what's important about this terminal phosphate group is that this bond right here, or excuse me, this bond right here that holds on or I should say attaches that terminal phosphate to the rest of the molecule, it is a high energy bond. And so when it's broken, a good amount of energy is released, and then it also takes the same amount of energy to reattach a terminal phosphate group. Think of an ATP molecule, I know this is a little silly, but think of an ATP molecule as kind of an energy savings account. When we need to make a withdrawal, when we require some energy for some task, we remove the terminal phosphate group, release some energy, use it. When we have, let's say, an excess of energy, we can store it in the form of that high energy bond that reattaches that third phosphate group onto an ADP molecule. Okay, a few words about enzymes. Enzymes are, protein molecules, and they are going to help cells speed up chemical reactions. Enzymes act as catalysts, and a catalyst speeds up a chemical reaction, and when we have a catalyst available, we require less energy to cause that reaction to go to completion. Now, all enzymes are proteins, but not all proteins are enzymes. And typically enzyme names end with ASE. Let's see what else I want to tell you. They are globular molecules. They have a very specific three-dimensional shape. If that three-dimensional shape is not maintained, then that enzyme is not going to be functional. Now, enzymes are, generally speaking, and keep in mind there are always exceptions, but generally speaking, enzymes are specific, meaning one particular enzyme can only catalyze one reaction. Enzymes are not consumed. They are recyclable, at least to a point. And another important thing about enzymes is that as they catalyze a reaction, there is not going to be a significant increase in the temperature of the environment, which is good for living cells, because if we raise the temperature above a critical point, they will, of course, perish. Okay, let's talk about enzyme components. Most enzymes are referred to as hollow enzymes, and they are going to consist of two parts. The part referred to as the apoenzyme is protein. Now, there will also be another portion making up this enzyme called the cofactor. And cofactors can consist of some different substances. For example, as it says in your notes, the cofactor might be a metal ion, such as, and I'm looking at the examples, you have copper, iron, magnesium, etc. Or the cofactor could be a complex organic molecule, which is called a coenzyme specifically. Examples, and we're going to talk about them in just a few minutes, include NAD and NADH, and these are charged molecules, and they are going to play a very important role in the movement of hydrogen ions and or electrons in a number of chemical reactions. Right now, the mechanism of enzyme action, well, it isn't clearly understood yet, but here's what scientists believe at this time. And we're going to have our enzyme molecule, of course, and remember I mentioned earlier it's got a very specific three-dimensional shape. There are areas on that enzyme molecule that are tailored, shaped specifically for the substrate to fit into the molecule. Kind of that lock and key analogy is what we usually use here. The substrate is the molecule that we want to change in some way. We might be breaking the substrate down to release some energy. We might be attaching another portion to it, for example. And so as you look at that diagram in your notes, you see that the substrate fits very neatly into the three-dimensional areas on the enzyme. And what we have when the substrate and the enzyme actually join together is what's called an enzyme substrate molecule. Now, this is a temporary intermediate state of being for both the enzyme and the substrate. Now, the enzyme's going to work its magic. It's going to make whatever changes, like the examples I mentioned a moment ago. And then once the reaction is completed, then the substrate will be released from the enzyme, and the enzyme will be available to participate in the same reaction again. All right, so what are some factors that are going to influence the activity of enzymes? Well, first of all, temperature is very important. Enzymes are going to have a very narrow range of temperatures in which they can function properly. If we go very much below that comfortable range, that's going to slow down enzyme activity. If we go too far above the comfortable range, it may actually denature the enzyme, and that may be temporary or it may be permanent. When I refer to denaturing of enzymes, I'm talking about either a temporary or permanent loss of that very specific three-dimensional shape. This, of course, is going to render the enzyme ineffective. Okay, so high temperatures, low temperatures. The pH of the environment is also critical. Very similar to what I just said about temperature. We've got a narrow range of pH in which the enzyme will function properly. If we become too acidic or too alkaline, we may temporarily or permanently denature our enzyme. And also, enzyme activity is going to be greatest when we have a high concentration of the substrate available to work with. All I mean by that is that when we have plenty of substrate available, then all of the available enzyme molecules will be working at the same time, so we're going to have a faster reaction rate. All right, so now another factor that is going to influence the activity of enzymes is going to be the presence of substances that are referred to as inhibitors. And you've got some nice diagrams in your notes here. We've got two types of inhibitors. First of all, are competitive inhibitors. Now, what those substances do is they actually get into that, that specific three-dimensional spot on the enzyme molecule that the substrate would normally occupy, and so just the space on the enzyme is filled, so the substrate just can't get in there, so the space has been blocked. And then non-competitive inhibitors, what they do is they bind to some other area on the enzyme molecule, but when that happens, the lock and key mechanism, the three-dimensional shape of the active site in the enzyme is going to be changed. Its conformation is going to be changed. And so that means that the substrate just can't fit in there. Now, in some cases, these inhibitors may have a permanent effect on the enzyme. In other cases, it may be reversible. Okay, let's change gear for a few minutes here and let's talk about some energy production methods. And I just want to kind of give you some highlights of some different mechanisms that microorganisms and other cells can use to produce energy. All right, so as it says in this section, we're going to look at some energy production methods. And what we're doing with these reactions is forming ATP molecules for use at a later date. And we're going to look at two categories of energy production methods. First of all are what we call oxidation reduction reactions. Sometimes they're called redox reactions. And then we're going to look at some different types of phosphorylations. Now, the term phosphorylation simply means adding a phosphate group to a molecule. So, for example, earlier when I was talking about adding that terminal phosphate group to an ADP molecule, what we've done is phosphorylate ADP. All right, so first on the list are oxidation reduction reactions, and we refer to these reactions as being coupled reactions. Now, what I mean by that is that when an oxidation is occurring within a cell, simultaneously a reduction is occurring, and I'll talk more about that in a few minutes. All right, now just want to see what you've got here. Let's go ahead and start with a definition of oxidation, and then we'll look at an example of an oxidation, and then we'll move on to reductions and then try to tie it all together. All right, so the definition that you've got in your notes is oxidation refers to the removal of electrons or hydrogen ions, and we're going to use those terms synonymously in this discussion. And this may sometimes involve adding oxygen to a molecule. All right, so let me do that again. Oxidation refers to the removal of electrons or hydrogen ions, okay? That's definitely going to happen in an oxidation. And when we do that, we may replace, or let me say refill the vacancy by adding an atom or more of oxygen at the same site on the molecule where we remove the hydrogen or the electrons, hydrogen ion. And the reason for that is, the reason for the possible addition of oxygen is that we need to satisfy the valence of all of the atoms involved. If it is an oxygen, it might be something else. All right, so if you look at this reaction, this is kind of the classic example of an oxidation, if I can get the darn thing to cooperate with me here. Okay, what we're looking at is a molecule of lactic acid, and I drew it out like this just so that you could see where all of the hydrogens were. Now, we're going to start with lactic acid, and we're going to oxidize it to produce a molecule of pyruvic acid. And so what we're going to do is, and I put a little asterisk here, by the two hydrogens that are going to be removed. So we're going to remove those two hydrogens, and I'm going to show you how coenzymes are involved in that reaction in just a couple of minutes. But those two hydrogens are going to be removed, so that's why I have minus 2 hydrogens here. And when that happens, we're going to have to form a double bond between our carbon and our oxygen there, again for valence satisfaction reasons. And in the process, we have formed a molecule of pyruvic acid. Okay. So that is an example of an oxidation. Oh, by the way, did you notice how I drew the arrow? I only drew a half of an arrow because this is only part, or this is, I should say, a reversible reaction, but we're only talking about the possibility of an oxidation with this example. Okay, so let's go ahead and turn a page and let's talk about reduction reactions. Now, a reduction is, well, just the opposite. Doesn't necessarily involve oxygen, but what's going to happen is we're going to add or see a gain of electrons or hydrogen ions to a molecule. And if you were to take, and I don't think I have anything to write with here, okay, let's say we drew the arrow in this direction, all right? So if we move from pyruvic acid to lactic acid, so what we would do to accomplish that is add 2 hydrogen ions to pyruvic acid. When we do that, we are now creating a molecule of lactic acid, and we have reduced pyruvic acid to form a molecule of lactic acid. All right, now let's take a look at what's happening to those hydrogen ions in these oxidation and reduction reactions. All right, now when I said earlier that oxidations and reductions were coupled reactions, here's what I didn't mean. I do not mean that when lactic acid is oxidized to form a molecule of pyruvic acid, that simultaneously pyruvic acid is reduced to form a molecule of lactic acid. Here's what I mean. Okay, now we can't just go plucking hydrogen ions off of molecules in the cell and tossing those hydrogen ions into the cytoplasm, because that would very quickly lower the pH to a point where the cell would perish, all right? So hydrogen ions, as we increase the concentration of hydrogen ions, we are increasing the acidity of the environment. So this is where our coenzymes are going to come into play. Let's just look at the top arrow here. So we're looking at oxidation of lactic acid to form pyruvic acid, all right? So remember, in order to do that, we're going to remove 2 hydrogen ions from our lactic acid molecule, and those 2 hydrogen ions, they're going to get incorporated into our coenzyme NAD. So up here I'm showing you NAD plus 2 hydrogens produces NADH2. Actually, that's not really correctly balanced, but let's just not get too upset about that right now. So let me just reiterate that as lactic acid is oxidized to form pyruvic acid, simultaneously our coenzyme NAD will be reduced, because it's going to, the NAD is going to pick up and incorporate those 2 hydrogen ions into itself. All right, so that would be an example of coupled reactions and oxidations and reductions. Okay, let's see where I want to go next. Okay, let's talk about some different methods that we can use to generate ATP. And by doing so, I mean phosphorylating ADP. All right, we've got 3 methods that I wanted to just briefly discuss. First of all is what we call substrate level phosphorylations. You've got a definition there. ATP is generated when a phosphate group is directly transferred from an intermediate metabolic compound to ADP. Here's what I mean. This simply involves, and coenzymes are going to be involved in this, but I'm not going to bother showing you exactly how they function. They're very similar to what I just showed you. So the example that you have in your notes is we've got a carbon compound that has a phosphate group attached to it, and that funny little squiggly line, that represents a high energy chemical bond. And then coenzymes are going to remove that phosphate group from our carbon molecule and are going to transfer it to our molecule of ADP. So what we have done is we have phosphorylated ADP to produce a molecule of ATP, and then the coenzymes are going to donate a hydrogen ion to that original molecule just so the valence requirements are satisfied. All right, next is what we call oxidative phosphorylation. And this occurs, as it says in your notes, in the plasma membrane of prokaryotic cells and in the inner mitochondrial membrane of eukaryotic cells. Now, in this situation, we're going to have a series of reactions of electron or hydrogen ion transfer. So what I mean by that is we're going to transfer hydrogen ions, let's just say hydrogen ions, from molecule 1 to molecule 2. When that happens, a little energy is going to be released. Then we're going to transfer the hydrogen ions from molecule 2 to molecule 3. When that happens, a little energy is released, and so forth. There can be a number of steps here. Now, every time we release a little bit of energy going through our transfers from molecule 1 to molecule 2, that energy can be used to perform a phosphorylation of ADP. All right, so this is how we're producing some energy that we're going to require to add that terminal phosphate group back onto an ADP molecule. All right, let's see, what else do I need to tell you about that? What I need to tell you is that when we're done with that series of transfers, the hydrogen ion or electrons are going to wind up bound to an oxygen atom. Okay, let's see. Next are what's known as photophosphorylations, and these occur in photosynthetic cells. Some bacteria, like for example some of the archaeobacteria, are photosynthetic. Photosynthetic cells in plants, for example, they work in this manner too. Now, what's different here is that our original source of energy would be light photons. So light strikes a photosynthetic pigment like chlorophyll, for example. Bacteriorhodopsin is another one, but just don't worry about the bacteriorhodopsin, just an example of a photosynthetic pigment we may find in a bacterium. So photons strike the photosynthetic molecule chlorophyll, and when that happens, that's going to liberate some electrons from the chlorophyll. Then those electrons get passed through a series of reactions exactly like I described in the oxidative phosphorylations. Two possibilities excuse me, for the fate of the electrons that are going through all of these transfers. First of all is what we call cyclic photophosphorylation. In that situation, after a number of transfers, the electron will be returned to chlorophyll, but at a lower energy content because we've harvested some of that energy through the reactions. And in non-cyclic photophosphorylation, the electrons do not return to chlorophyll. They'll be incorporated into some other molecule. All right. Let's see what we've got left. Okay, let's see, a little bit of reading in the textbook. I'd like you to read the section on carbohydrate catabolism in your textbook and be able to define, and there are a list of bullet points, terms that you need to be able to define. And next we're going to talk about nutritional patterns. Okay, the last topic in this chapter would be nutritional patterns. And what we're looking at here is separating microorganisms, actually I should say separating all organisms on planet Earth based upon two criteria. One is how do they produce their energy? For example, do they use oxidation reduction reactions, or do they use one of the varieties of phosphorylations that I talked about previously? And then the second criteria is where do they get their carbon? Some bacteria, some organisms will strip carbon from a carbon dioxide molecule. Others may harvest carbon from an organic molecule, such as a sugar, for example. And so, first of all, we look at those two criteria separately. We look at energy source, we look at carbon source, two possibilities for each of those. For energy source, we have what are referred to as the phototrophs, and those are organisms that use light as their original source of energy. And the second category are known as chemotroph. Troph means to feed or feeder. And chemotrophs, they are going to utilize oxidation reduction reactions to produce the energy that they need for example, phosphorylating ADP to form ATP. Then we've got also two possibilities for carbon source. First of all, are what we refer to as autotrophs. Those organisms harvest carbon from carbon dioxide molecules, and heterotrophs, they're going to require some organic molecule to obtain their carbon. All right, and then if you go ahead and you merge those two categories together, you form four new categories. And by the way, this is this last part here, the four categories: phototrophs, photoautotrophs, photoheterotrophs, chemoautotrophs, and chemoheterotrophs. This is one of your possible essay questions. So let's go ahead and run through them together. Photoautotrophs, all right, they use light for energy and they remove carbon from carbon dioxide molecules. And you can see some examples there, a lot of examples there. Algae, plants, like, all plants, the cyanobacteria, and a rather unusual group of bacteria referred to as the purple and the green sulfur bacteria. That means they have either a purple or a green photosynthetic pigment, and they are able to metabolize sulfur-containing compounds. Photoheterotrophs use light for energy and organic compounds for harvesting carbon, and not so many in this category, purple and green non-sulfur bacteria. So they have those pigments that I mentioned a moment ago, but they are unable to metabolize sulfur-containing compounds. Chemoautotrophs, they're going to use inorganic compounds, oxidation reduction reactions of those compounds to produce energy and carbon dioxide to harvest carbon from. Many species of bacteria fall into this category, and some examples of inorganic compounds they might use include hydrogen sulfide, sulfur, nitrite, hydrogen, ammonia, and iron. And then the fourth and final category are chemoheterotrophs. And in this case, energy and carbon are usually going to be derived from the same molecule, more often than not an organic molecule. And we can kind of break that category down further. Not all of the examples fit into this, but into saprophytes, which are organisms that decompose organic material, and our parasitic organisms, which of course make a living off of a live host, fall into this category as well. Examples include all animals. So if anybody asks you, you happen to be a chemoheterotroph. Many species of bacteria fall into this category, all of the fungi, and most of the protozoans. The exceptions there for protozoans is there are some protozoans that have photosynthetic pigments, and so they would be autotrophic. All right, you guys, this wraps up the metabolism chapter. I will look forward to seeing you soon.